Hi, my name is Afaf, and this video is about the evolution of endorphins in the human brain. What is it about us humans that make us so different? Even with the many similarities to other primates, humans appear to have superior cognition. About 30 years ago, geneticists Mary Claire King and Alan Wilson proposed the idea that gene regulation is the key to understanding the reason behind these differences. New studies of evolutionary changes are focusing on the regulation of a gene involved in perception, behavior, and memory, suggesting that the two scientists were partially right. More and more studies are slowly revealing how selection on a regulatory element and certain brain genes have been favored during the evolution of humans. Although it is not yet clear exactly what behavior traits were favored in this case. Recent studies are focusing on the gene that codes for the protein prodenorphin. Prodenorphin is an opioid polypeptide hormone involved with chemical signal transduction and cell communication. The gene is expressed in the brain. It is a substance from which a number of endorphins are made. Endorphins are involved in learning, the feeling of pain, social attachments, and bonding. Prodenorphin gene is controlled by a promoter region that is just upstream from the gene's coding region. Studies had highlighted a 68 DNA base pair segment of the promoter. This promoter differs among humans who may carry one or up to 4 copies of it. It is not clear yet how the number of copies or the length of the segment affect the gene function, although some specific variants have been linked to schizophrenia, cocaine addiction, and epilepsy. In one study I found very interesting, scientists sequenced the promoter DNA from 74 human chromosomes as well as 32 chromosomes from 7 other different primates, including chimps and gorillas. None of the non-human primates had more than 1 copy of the 68 base pair segment. All humans' segments had 5 DNA mutations not seen in the other primates. The researchers from this study concluded that the pattern shows evidence of natural selection acting on the human lineage after it had split from the chimp line about 5 to 7 million years ago. But the question to be asked is, does the difference in promoter actually alter the gene expression? To answer this, the team of researchers introduced either the chimp or human 68 base pair segment into human neural cells. The answer was yes. The human segment induced a 20% greater expression of the prodenorphin gene than did the chimp segment. The evolution of gene regulation shows that regulatory mutations as well as structural mutations contributed to human evolution. Prodenorphin is important in human biology. There is a strong suggestion that the evolutionary changes in its regulation may have helped set chimps and humans apart. But this very reasonable hypothesis still awaits the answer to the most important question of all. Why were these changes favored by natural selection? And until this question is answered, it is nothing but a very beautiful hypothesis.